Welcome friends. Today's topic is non-tuberculous mycobacteria. So these are the objectives of the today's lecture. First, we will discuss the Reynolds classification of non-tuberculous mycobacteria. Then we will see the clinical importance of the non-tuberculous mycobacteria, and at last we will discuss the laboratory diagnosis of this type of organism. So first we will talk about the non-tuberculous mycobacteria, which is also called the atypical mycobacteria or mycobacteria other than tuberculosis, that is called MOTT. This non-tuberculous mycobacteria are diverse group of mycobacteria that are isolated from the birds, animals and environmental sources like soil and water. Some of the species of the mycobacteria are saprophytic mycobacteria, which do not cause any disease in humans, while the majority of the member of this group are opportunistic pathogen, which can cause the disease in the patient with lower immunity. So here is the Reynolds classification. This classification is based on the pigment production and rate of the growth. So these are the this is the classification. So this classification having a 4 broad groups. Among this, the first one is a photochromogens. Photo means light. So the photochromogens having a property like it produce the pigment only in the presence of light. The member which is important of this group is M. marinum and M. cansaceae. Then the second group is scotochromogens. That means it produces pigments both in light as well as in a dark. The most important member of this group is M. scrofulaceum. Then the third group is non-photochromogens, means that it does not produce the pigments. The important member of this group is MAC, that is Mycobacterium avium intracellular complex, that is MAC, that is MAC. And the other important member of this group is M. ulcerans. Then finally the fourth group, which is a rapid growers. Rapid growers means it can be grow within a 1 week. The important member of this group is M. colony. Let's talk about the clinical manifestation. So here is the list of disease that can be caused by the non-tuberculous mycobacteria. So first we will talk about the lymphadenopathy, which can be caused by the Mycobacterium avium intracellular complex, that is MAC, and Mycobacterium scrofulaceum. In the skin lesion, among the skin lesion there is the first is post-trauma abscess, which can be caused by the Mycobacterium chelone. Then the swimming pool granuloma, which is also called the fish tank granuloma, which can be caused by the Mycobacterium marinum. Then the Buruli ulcer, which can be caused by the Mycobacterium ulcerans. Then the respiratory disease, this disease is caused by the MAC, that is Mycobacterium avium intracellular complex, then M. cansaceae, then the M. xenopi, and other members of the group. And at last the disseminated disease. Among this, some are related to AIDS and some are non-related to AIDS. So the AIDS, the MAC can be caused the both type of disseminated disease. Then we will talk about the Mycobacterium avium intracellular complex in detail. So this complex is made up of Mycobacterium avium and Mycobacterium intracellular. So this is the complex of these two because it cannot be differentiated. This MAC is non-photochromogens, that means it does not produce the pigments. Then it can be caused the mostly opportunistic pathogen, mostly in HIV infected people. So when the HIV infected people having a less than 50 CD4 T cell count, then the chances of this infection of this MAC will be increased. MAC can cause a various manifestation. Among this, lymphadenitis, which can be shown in picture, then respiratory infection and disseminated disease are the most common ones. Then we will talk about the scrofula. The scrofula is a disease which can be caused by the Mycobacterium scrofulaceum. This Mycobacterium scrofulaceum is a scotochromogens. That means it can produce the pigment in both light as well as in the dark. It can cause the scrofula. In scrofula, patient comes with the cervical lymphadenopathy, which can be seen in the picture. Mostly the children are affected with the scrofula in compare of adults. So let's talk about the swimming pool granuloma. The causative agent of the swimming pool granuloma is Mycobacterium marinum. Swimming pool granuloma is also called the fish tank granuloma. Mycobacterium marinum is a photochromogens. That means it can only produce the pigment in the presence of light. When the patient is associated with the aquarium, then he gets the fish tank granuloma. When the patient is associated with the swimming pool, then he gets the swimming pool granuloma. Patient comes with the papules or ulcer, which is called the granuloma, and the tendonitis. When patient is associated with the swimming pool, then the most exposed site of the body is knee, elbow, and feet. While in the case of fish tank granuloma, the most common site are hands and fingers, which can be shown in the figures. Here there is a figure of fish tank granuloma, a patient having a papule or ulcer. Mycobacterium marinum can be grown only at 33 degrees Celsius, not at the 37 degrees Celsius. And this Mycobacterium marinum enters through the minor troba, where where the patient develops the different papules or ulcer, that is called the swimming pool or fish tank granuloma. Then we will talk about the Buruli ulcer. The causative agent of this ulcer is Mycobacterium ulceram. This is called Buruli ulcer because it derived from the Buruli district of Uganda, where there is a large outbreak had occurred. It is a non-photochromogens. That means it does not produce the pigment. It is a waterborne disease. Here patient comes with a painless nodule, which will be seen in the picture. This nodule becomes the necrotic later. Patient also comes with the osteomyelitis and the limb deformities. This Mycobacterium ulceram produces the exotoxin, which is called the mycolacton. This toxin may be involved in the pathogenesis of the disease. Mycobacterium ulceram having a narrow temperature range for growth, that is 31 to 34 degrees Celsius, and it grows slowly. Colonies will be appear into 2 to 4 weeks. Then the chronic pulmonary disease, which can be caused by the Mycobacterium cancersi. This Mycobacterium cancersi is photochromogen. The source of this infection is from the soil, water, and milk. This chronic pulmonary disease, which is produced by the Mycobacterium cancersi, is resembling a tuberculosis. Then the risk factor: old person with pre-existing lung disease or impaired immune response are more prone to this type of infection. Organism grows well at 37 degrees Celsius on algae media, where it produces the yellow-orange pigment that can be seen in the picture. Then at last, the laboratory diagnosis. First, the specimen collection. So the specimen can be collected depending on the type of infection. So there is a sputum, pus, lymph node aspirate, biopsy, etc. Specimen will be collected. Then we will do the microscopy. In microscopy, we will do the acid fast staining. In this, there is a red acid fast bacilli, which is similar to Mycobacterium tuberculosis, but we have to differentiate it. So we will do the culture exercise and identification of the non-tuberculous mycobacteria. So for the culture, we will do the culture on LJ medium, and we will put the LJ medium into the incubation in light and dark separately for differentiation between the photochromogens and scotochromogens. Then identification. Identification is done by the different methods. Among these, most important and most commonly used method is MPT64 antigen. This MPT64 antigen test is a rapid test based on the principle of immunochromatography. Uh, in the case of Mycobacterium tuberculosis complex, this test becomes the positive, while in the case of NTM, that is non-tuberculous mycobacteria, the NPT64 antigen test becomes the negative. Then we will also do the newer methods like MALDI-TOF and PCR. These methods currently preferred and for the species differentiation of non-tuberculous mycobacteria. Then we will also do the biochemical reaction for differentiation of the Mycobacterium tuberculosis from atypical mycobacteria. So niacin test, nitrate test and pyrazinamidase test is positive in the case of Mycobacterium tuberculosis. Then Mycobacterium ulceram and Mycobacterium marinum can be grown at 32 and 33 degrees Celsius respectively, while the Mycobacterium tuberculosis can be grown at 37 degrees Celsius. The catalase test comes positive in the case of atypical mycobacteria, while Mycobacterium tuberculosis gives catalase test negative. Some of the species of mycobacteria can be grown on the McConkey's agar, while the Mycobacterium tuberculosis gives the it cannot grow on the McConkey's agar. So this was all about the non-tuberculous mycobacteria. Thank you.